They don't like. I was not sure where to go, and then you can. Yeah, so it is such a.
Yeah. Meetings are an hour.
Thank you very much.
It's time to discuss. Other person's design.
What he was describing, and you also mentioned. He's all there.
Oh, this is great.
I like
Can you hear me now? Did anyone in the
Yeah,
Can anyone on Zoom hear us?
it's kind of like.
Can you guys hear us on Zoom? Give me some indication.
Can you
hear us okay, Jen? Okay.
Yep, I can hear you now. Perfect.
Oh, wonderful, wonderful, perfect.
Yeah, thank you. Yeah, I couldn't hear you first, but yeah, I think you pressed something and now I can hear you.
Brilliant, brilliant. Thank you. So what we'll do is we'll have a little introduction and for your actual presentation. So you can share your screen. Start it right before you talk, either way.
Perfect. Let me bring it up.
Yes.
So, welcome everybody. My name is Pablo Castillo. I'm Professor of Optics here at the Institute of Optics at the University of Rochester. And it's my pleasure to introduce Jennifer Dionne today. She is the Senior Associate Vice Provost of Research Platform Shared Facilities and Associate Professor of Material Science and Engineering and Radiology at Stanford. Jen received her PhD in applied physics at Caltech, by Harry Atwater. And BS degrees in physics and systems and electrical engineering from Washington University in St. Louis. Prior to joining Stanford, she served as a postdoc researcher in chemistry at Berkeley, advised by researchers. Jen's research develops nanophotonic methods to observe and control chemical and biological processes as they unfold with nanometer scale resolution, emphasizing critical challenges in global health and sustainability. The work has been recognized with many awards, and I can cite among them the Alan T. Waterman Award 2019, NIH Director's New Innovator 2019, the New Inventor Fellowship 2017, the Emerald Young Investigator Award 2017, the Adult Long Medal 2016, Long Foundation Fellowship 2015, and the Early Career Presidential Award against Scientists and Engineers in 2014. He also was featured on Oprah's list of 50 things that will make you say, wow. So thank you very much, Jennifer, for having for being here today with us. It's an honor, and the stage is yours.
Great. Thank you so much for the introduction. It's a pleasure to be presenting this on Zoom. I realize Zoom is not as fun as in person, but hopefully when the weather gets a little nicer and there aren't so many strains of viruses going around, I can visit you in person, or you all are welcome to come visit us at Stanford. There does seem to be a bit of background noise, so I would ask that if you're joining by Zoom, you can perhaps mute yourself, because I hear about 4 different conversations going on in the background. Today, I wanted to share with you some of the work that my lab has done on developing nanophotonic platforms for environmental and ecosystem health monitoring. And this work would not have been possible without some really incredible collaborators, especially those who are in Stanford School of Medicine and also in the Stanford Dorr School of Sustainability. And some of the work that I'll be presenting on was also recently translated by a spinout from my lab called Pumpkin Seed. So you'll get to hear a bit of the work happening there. So I thought I'd start off with a bit of a pop quiz. I know it's a Monday morning, so not the best time for quizzes, but if we were to take a milligram sample of biological material, and for now we can assume that maybe it's just made of DNA, how much data do you think would be stored in a milligram? And feel free to either shout out an answer if you're on Zoom, kind of guess, in bytes, how much data we could store in a snowflake size worth of, say, DNA.
3 terabytes.
Awesome.
2.5 terabytes.
A terabyte. 1 terabyte.
10
Great. Okay.
terabytes.
I'm hearing lots of terabytes, which are good guesses. It turns out it's actually an exabyte, so 10 to the 18th byte, which is a pretty remarkable amount. More data than that stored by Microsoft, Google, Amazon, and Meta in any given day. And yet, the rate at which we can read these biological samples, or the data that's contained within these biological samples, is still relatively slow. So the fastest biomarker that we can read right now is DNA. We can sequence DNA at rates of about 400 bytes per second. We compare that to an average internet speed, which can give us fiber optic data at rates of 100 megabytes per second. You can see that even DNA, which is DNA sequencing, which has generated tons of data, still is far behind what we can do with state-of-the-art photonic technologies. And then if we think about kind of the proteins and millions of cells and metabolites that might be in a small compact size of any biological sample, whether it's from a human, an animal, the environment, we're just not getting the data we need to be able to understand ecosystem and environmental health. And part of the reason why it's so hard to extract the data is because of the workflow we have. So if we look at kind of the tools that we have to analyze biological samples beyond just DNA sequencing, if we want to look at cells or proteins, we're limited by culturing the cells, or by doing like ELISA type assays, or by using mass spectrometry, where throughput is really a challenge with mass spec. Generally, you can get through about 12 samples a day. And that workflow doesn't allow us to have either high throughput or longitudinal analysis of both population, personal, and ecosystem level health. So today we'll talk about how we can develop new technologies to more rapidly read kind of the data within biological samples by merging silicon photonics and machine learning with biological samples. And I want to share with you, first of all, how we develop kind of the silicon chip technology, and then we'll use those silicon photonic chips for amplification-free gene and metabolite detection. So focusing first on kind of small molecule fragments. And those are labeled techniques where basically we develop our highly multiplex silicon chip that has millions of sensors per centimeter square, and we can target particular biomolecules that we want to look at. And then in the second part of my presentation, I'll kind of shift over to how we can use a similar platform, but for more label-free detection, in particular relying on the vibrational spectra of molecules to be able to detect both whole cells as well as proteins where you may not want to take a targeted approach, but just kind of understand what more broadly could be in the sample. And then in the last portion of my talk, I'll share with you how we're deploying these techniques both to detect pathogens in wastewater and then also to understand harmful algae blooms in ocean water by collaborating with both kind of local wastewater facility management systems here in the Bay Area and also with Monterey Bay Aquarium Research Institute to be able to deploy some of our chips onto these autonomous underwater vehicles. And ultimately what drives our work is to help enable the transition from a reactive and crisis-based approach to health monitoring to a more proactive and solutions-based approach. So University of Rochester is, I think, one of the leaders in optics and optical devices, so it's probably no surprise that for fast reading of genes, proteins, metabolites, and whole cells, we're going to rely on optics, where we have the advantage of essentially computing at light speed. And historically, these optics-based sensors have relied on basically a shift in the refractive index or a shift in the scattering spectra that emerges from kind of target binding in the vicinity of whatever sensor you're using. And probably we've all used at this point an at-home SARS-CoV-2 test. Perhaps some of you have used an at-home pregnancy test. Those basically give us different strips that light up usually pink when the antigen is present, and that's based on kind of how binding changes essentially the refractive index of various nanoparticles, and hence their optical scattering spectra. So what we wanted to do is kind of take some of the insights from the many decades of work that have been done in optical sensing technologies, and first of all, figure out how we can improve the detection capabilities or the sensitivity and be able to enable sub picomolar or at least femtomolar scale resolution or detection limits. We also wanted to enable multiplexing, so ideally have a chip that can detect a large number of molecules on a given chip, and then also scale for greater throughput, so that way we could look at more samples per day. So we took inspiration from nature's light harvesting organisms to be able to design sensors that meet all these criteria. So high sensitivity, high resolution, high throughput. And I was lucky enough to be able to recruit an evolutionary biologist into my lab. She's a postdoc, Cody McCoy, actually on the job market this year. And she's interested in studying kind of nature's light harvesting organisms like corals and heart cockle clams that have evolved essentially over millennia to be able to precisely focus light onto their photosynthetic symbiont, so that way they can control the amount of light that's going to the systems that are essentially helping them to photosynthesize and produce energy. And unfortunately, they have grown almost too good at this, where some of these organisms are now starting to bleach. But Cody put a lot of effort in her postdoc in my lab studying, for example, the heart cockle clam. And if you look at these clams, their shells are heart-shaped, hence the name. So here you can see a side view. And essentially, they tend to bury themselves in the sand. So the top face of the shell is exposed, the bottom face is in the sand. And if you look pretty closely at the shell, you can see there are a lot of little grooves and divots. And what Cody did is do kind of laser scanning tomography of the grooves in the heart cockle clam. And then you can see here the output of one of those tomographic scans or topographic scans. And then she put that into a full field simulation in COMSOL and showed that by virtue of the structure of this particular lens shape, you can actually get focusing of the ultraviolet light at a depth that's a few hundred microns below the minima of the lens. And this is actually where the light is now getting focused onto the photosynthetic symbionts. And then what you can also do is image kind of straight through the shell. So she put a radicule behind it, basically this ruler. And you can see that when you are focused on the bottom side of the lens, you can kind of clearly see the image. And part of this focusing, of course, comes from the particular shape of the lens, but she also took a cross section. So this is now in this grayscale SEM looking kind of in a cross-sectional view of the shell. And you can start to see these fiber optic bundles that have diameters of about 500 nanometers up to a micron. So it's pretty neat to see how in nature, nature has evolved basically to have both fiber optic bundles and lenses that can both kind of direct the light to where it needs to go, as well as focus the light basically onto the organisms. So in our lab, kind of taking inspiration from this structure, we wanted to make kind of combined waveguides or fiber and lenses structures that could both focus light in the near field where the molecule is going to be, but then direct the light to the far field, in other words, to our camera. So that way there would be a one-to-one correlation between where each molecule was, or where each sensor was, and where each pixel detector was. So basically, to be able to create these structures, let's just first of all consider a series of silicon waveguides. And here the waveguides are going into and out of the page. You can see the center of the waveguide indicated by each pink dot. And then this is just a full field simulation where light is coming from the bottom. And you can see in this waveguide array of silicon waveguides that are each about 210 nanometers wide, light just gets transmitted straight through. But now if we start varying the width of each neighboring waveguide, for example, having a silicon waveguide that's 210 nanometers next to a 220 nanometer waveguide structure, and then next to a 400 nanometer waveguide structure, you can see each of these waveguides that is into and out of the plane kind of acts like a dipole scatter. And each of those dipoles is slightly out of phase. So when we wind up stacking them all next to each other with this 3-bar unit cell, you can see the phase difference basically gives rise to beam steering in this case. So light here is steered to 45 degrees to the plus first diffracted order. You can also, instead of considering a linear phase profile, essentially have a hyperporoidal phase profile of the silicon waveguide bars, and that leads to lensing. So the cool thing is that all of this is happening now in a submicron thick structure where we don't have any geometric patterning of the bars. Instead, we're just varying the phase between each of the neighboring waveguides or each of the neighboring bars to be able to get beam steering or lensing. So we're going to use that as kind of our building block for how we can get structures that focus light to the far field. But then I mentioned that each silicon structure is essentially a waveguide into and out of the plane. And basically, if we plot the dispersion, so basically the free space wavelength as a function of the guided mode wavelength, we can see each bar basically has its own unique waveguide modes. And if we take, for example, the waveguide mode that's in this thickest bar, the 400 nanometer thick bar, and we plot at a free space wavelength of 1500 nanometers where we're exciting the waveguide, you can see it has a guided mode wavelength of about 520 nanometers, and the light here is strongly focused. There's also a waveguide mode that corresponds to that middle 240 nanometer width bar, a waveguide mode that corresponds to the thick bar again. This is a higher order waveguide mode. And then here's the waveguide mode of our thinnest bar. And these waveguide modes are normally inaccessible by free space light. So light normally is just going to pass through these waveguide structures and then have, say, that beam steering or that focusing that you saw in the previous slide. But what we hypothesized and what's been shown in a number of papers now is that we could utilize the guided mode resonances to be able to get extremely sharp resonances in our diffraction spectra. So basically to be able to instill a high quality factor, and that high quality factor is what gives us high sensitivity in our sensing. So here's that 3-bar waveguide structure where we have one bar with a width of 210 nanometers, one bar with a width of about 240, and another thicker bar that has a waveguide width of 400 nanometers. And I showed you earlier how when light is incident from the bottom, it gets beam steered to that plus first diffracted order. But if we take one of our bars and we insert notches into it, those notches basically take the free space light and couple it into a waveguide mode. So that way we have now interference between the light that's directly transmitted through each of these bars, as well as that waveguide type mode. And with that interference, we wind up with an extremely sharp dip in the scattering spectrum. So if we look at the diffraction efficiency to that plus first diffracted order, you can see here's a sharp dip with a quality factor on the order of about 8,000. And what that means is that we wind up strongly enhancing the incident electromagnetic field, in this case by about 100x compared to what we would get without the notches. So why is this very sharp scattering spectrum important? Well, if we go back and revisit those optical sensors that I showed you at the beginning of my talk that we use, for example, in at-home COVID tests, we know that there's a high degree of false negatives, and that's because they have a relatively high lower limit of detection. So because most at-home sensors or optical sensors have a low quality factor, that means they have a very broad scattering spectrum. And when there are molecules, for example, COVID antigens that are binding to our sensor, basically it takes a large number of molecules to be able to get a measurable shift in the scattering spectrum to be able to see that color change. But if we take a high quality factor resonator like the one I just showed you that considers the interference between a waveguide type mode and a standard Me, as in Gustav Me resonance, then what we can do is basically instill a sharp scattering resonance in the diffraction spectrum. And that means when there are only a few molecules landing on our sensor, we can get a much sharper change in the scattering spectrum and therefore have more sensitivity to just a few molecules that are landing in the vicinity of our sensor. So this is basically the design principle of the structure we're using. We're going to be taking structures that combine Me, Gustav Me resonant modes with waveguide modes to be able to get sharp scattering spectra. But then we also design the structures in such a way where they can focus the far field light to particular detector pixels. So if we want to have dense multiplexing, we need to go from this infinitely long waveguide type structure to something that's much shorter. And in most of our previous work, what we had considered our long waveguide structures with lengths of about 100 microns, and those theoretically gave us Qs of about 100,000. In fact, they could be quite a bit higher with some engineering. But as you start truncating down the waveguide length, then you get scattering out of the end facets of the waveguide, and that gives rise to an additional radiation channel that basically significantly diminishes the quality factor. So that's not good. If you want to be able to densely multiplex, you need to be able to pack as many of these onto a chip as possible. So ideally we can have short waveguides that don't compromise on the quality factor. So what we did is essentially consider a shorter waveguide, where here we have this cavity section where we have kind of our notched bars. And then what we do is insert photonic crystal mirrors that are essentially at the end of each waveguide structure. And then at the end of each photonic crystal mirror, what we have is essentially a padding region. So the photonic crystal mirrors help to back reflect the light back into this high quality factor waveguide, or this cavity region. So that way we can make a short waveguide look as if it's an infinitely long waveguide by virtue of putting these mirrors at the end. And my student Varun has done quite a bit of work optimizing these structures. I'm happy to go into their design during the Q&A if you're interested. But what he did is essentially consider different photonic crystal taper geometries. So in the middle, we have this waveguide region. And then we have photonic crystal sections that are linear or quadratic or cubic or quartic, all the way up to 6-degree polynomial tapers. And what he did is calculate how the quality factor varied as a function of the order of the polynomial for the taper. And now, like I just showed you, we know that that quality factor drops down by a factor of at least 10 to 100x when we have no photonic crystal. But here what he was able to show is that when you have various notch depths, in this case delta D, that allow for your free space light to couple into that waveguide mode, for the appropriate order of the polynomial taper, usually 3 to 4, you can get quality factors that are approaching hundreds of thousands or exceeding hundreds of thousands, approaching millions. And what he did is characterize kind of the best structures that we could make in Stanford's clean room at the time, which have asymmetry factors or notch depths closer to 100 nanometers. So I think with state-of-the-art fabrication, we could probably boost this quality factor up. But what he did is basically vary the order of the polynomial taper for some of these larger notch depths, which are predicted to have quality factors in the thousands. And he was able to achieve these nice sharp scattering spectra where he was achieving quality factors of about 2000 in some of his best devices. So the cool thing about these finite pixel structures, or as we call them, finpics, is that they have high quality factors in the near field that strongly concentrate light, but they're also able to focus that light to the far field. So you can kind of view them as nanoscale lenses that focus light to where the molecule will be, but then they also direct that light to the detector pixel. So in this talk, we essentially rely on two types of finite pixel structures, one of which I just showed you has that cavity region or that waveguide region. And because we're doing molecular sensing, we actually moved away from a continuous waveguide to more of a fully slotted waveguide. And that's because that allows us to push the field outside to where the molecules will be binding. And you can see here that this particular structure has quality factors in the thousands and allows us to get electric field enhancements in this cavity region that are over 2500. So this particular structure allows us to achieve kind of cellular scale sensing. So we'll use this structure largely for detecting whole cells. We also have some designs that allow for more molecular scale sensing, where what we can do is essentially put a notch into our waveguide, and that allows us to dramatically shrink down basically the mode volume from just a few wavelengths to less than 1/100th of the wavelength. So here we have attoliter scale sensing volumes that allows for very high resolution molecular scale sensing. And with these finite pixel structures, we can integrate many high quality factor biosensors onto a single chip. So on a 1 centimeter square chip, which is the size on which we make most of our sensors, in the clean room, we can pattern about 3 million sensors. And for any of you in the audience who might have done like bio or wet lab work before, on this small 1 centimeter square area, we're able to pattern the equivalent of over 31,096 well plates. So if you were to try to do all of these biological assays in a well plate format, you would essentially need the size of a basketball court of well plates to be able to have an equivalent number of assays as each of these nanofabricated chips. So here's a schematic of one of our chips. What we can do is broadly illuminate the chip from the back side. And then, like I said, you can think about each of these sensors as being high quality factor resonators that are addressable by free space. And then they kind of focus light in the near field, but also direct it to particular CCD pixels. So we can do broad area imaging of each sensor. And since we have so many resonators on a chip to be able to take advantage of all of those for multiplexing, you need to develop chemistries that can basically attach biomolecules to individual pixels. So my student Fariha Safir, who is co-advised with Pierre Curie-Jacob, is an expert in acoustic droplet ejection, and she was able to develop a bioprinter that could take open pools of liquid, for example, of different chemical reagents, and basically put those on top of piezoelectric transducers, where the size of the droplet that was ejected was proportional to the piezoelectric frequency that was applied. So here you can see the droplet being ejected from a pool of single-stranded DNA. She's done stroboscopic imaging, so you can see the necking of basically the droplet as it's coming out of the water. And then each droplet diameter is about 10 microns across. So depending upon, like I said, the frequency, you can control the droplet diameter. Generally, commercial systems are working at these slightly lower frequencies, but by moving up to megahertz rates, you could essentially print 10 micron scale droplets. And here she printed the Stanford logo. And here you can see basically printing over individual resonators in kind of a checkerboard pattern. So this is a bright field image where you can see every other resonator has been patterned. In bright field, I don't think it shows quite as clearly as it does in fluorescent. So here what she did is basically use bioprinting to be able to print down a protein, in this case a Y-shaped protein or an antibody. And then what she did is flow in over the entire chip a secondary antibody that was fluorescently tagged. So here you can see that secondary antibody, even though she introduced it to the entire chip, only binds in the region where you have chemical specificity for that particular antibody. And the cool thing is you can see actually how uniformly the surface gets coated when you're using bioprinting. So the, I think, really cool thing about bioprinting is that by controlling the humidity and the drying conditions of each droplet, you can actually get much better uniformity of packing of surface monolayers than you can get with traditional surface chemistry approaches. So what we do now is we take our chips that are locally functionalized to be able to target either genes or proteins or metabolites. And then, like I said, we can broadly illuminate the backside of the chip as we flow in different analyte solutions. So here what I'm doing is showing you imaging of our chip. So each bright spot that's lighting up is a different sensor. And what we're doing here is using a tunable laser to sweep the frequency that's going over our chip. And you can see different sensors are lighting up at different wavelengths. So if we look at individual frames, each corresponding to a distinct wavelength, you can see some sensors are bright at lambda 2, whereas other sensors are bright at lambda 1. And we can use these hyperspectral images to basically individually reconstruct spectra of each of our sensors so we know what's landed where. And from these individual spectra, we can get quality factors that are in either buffer or aqueous solutions, including ocean water, routinely in the thousands. Our record is about 14,000 right now, based mostly on what we can fabricate in Stanford's clean room. So to kind of summarize the physics before I get into some of the biological applications, if we look at the landscape of previous optical biosensors like the home COVID test, they've normally relied on either plasmonic particles or dielectric Me resonators. And what those structures give us is the ability for very good kind of spatial and diffractive control. Many times they scatter much like electric dipoles or magnetic dipoles. But the quality factor is usually quite low, meaning that the scattering spectrum is very broad. So it's hard to get a very sensitive device that can detect below, for example, nanomolars of analyte concentration. Then as we move up to either higher order multipoles or kind of these bound state and continuum modes, or to photonic crystal cavities and ring resonators, we can get much higher quality factors and much more sensitive devices, but the scattering tends to go in every which direction. So we no longer would have that one-to-one correspondence between what's happening on the chip and how the light is being steered or directed to particular detector pixels. So generally, as we increase the quality factor, we wind up reducing the degree of spatial or diffractive control that we get. And what our work has shown is that this trade-off between spatial control and temporal control is by no means fundamental. We can basically overcome that by developing what we call high quality factor metasurfaces that both strongly confine light but also steer it. And I think the advantages of these high quality factor metasurfaces is that they have a fairly lightweight and kind of flexible form factor, so they can be applied to a number of different environments. And we also just use conventional CMOS fabrication processes, so we can fabricate these on 8 or 12-inch wafers if we needed to. So let's put these high quality factor metasurfaces to work. And what I'll share with you is, first of all, 2 labeled techniques. So in other words, how we can use that bioprinter to put down either aptamers or biomolecules that are specific to the target we want to detect. And then we'll kind of shift gears and talk about some label-free techniques, in particular vibrational scattering, in order to be able to detect proteins and peptides as well as bacteria. So on the nucleic acid side, what we did is essentially take our silicon chips, we functionalize them with a silane layer, and then what we do is add a thiolated DNA probe that has a sequence that's complementary to the sequence we want to detect. And in this case, since the work was done about 1.5 years ago at this point, we were interested in detecting various SARS-CoV-2 gene fragments, including the E gene of SARS-CoV-2 and the open reading frame or ORF gene of SARS-CoV-2. So here what we did is use hyperspectral imaging of our chips, where we're collecting resonances from many different sensors simultaneously. And as we flow in the different biomolecules to be able to functionalize our surface, so going from the bare silicon chip to the silane coated chip to the chip that has the probe single-stranded DNA on it, to then flowing in the target, you can see there's roughly a 0.5 to 2 nanometer shift in the scattering spectrum with each additional molecular monolayer that's added to the chip. And that agrees quite well with what we simulated for these particular structures. And then if we plot the resonances of all of our sensors, you can see we have roughly about a 0.5 nanometer shift as we go from the probe single-stranded DNA to the target DNA binding to the chip. And you can see each of these dots corresponds to an individual resonator. So in this case, we're able to measure roughly about 100 resonators simultaneously. The cool thing about this particular design is that because we're interrogating biomolecules in one of these finite pixel structures that's only about 10 microns by a few microns across, we can benefit from like the extremely fast kinetics. So within just 5 minutes of adding the target, you can see that the signal is saturating. And again, here we're measuring over many resonators simultaneously. So it's a very steep turn on and saturation when we introduce a biological sample. And because we're able to do liquid measurements, we could extend this not just to nucleic acids that were in buffer, but also in clinical nasopharyngeal swabs, working in collaboration with the clinical virology lab at Stanford. So they were able to send us samples of nasopharyngeal eluates. And basically with our chip, what we do is introduce a negative control where we know it's been confirmed negative by PCR. Then we flow in buffer, and that basically cleans the chip off. So you can see no statistical wavelength shift with these negative samples. And then when we introduce positive samples, we see that there is that 0.5 nanometer or so shift, and that remains even when we flow in buffer. That should basically clean off anything that's non-specifically bound to the surface. And what we found in terms of our concentration studies is that we could optimize the single-stranded DNA that was on the surface to ensure that there were no hairpin loops that were formed at room temperature. And by optimizing kind of the probe that was positioned onto our surface, we could look at the wavelength shift as a function of the concentration of the single-stranded nucleic acid that was in the solution, and we could detect below 8 femtomolars. So right now that's kind of our best limit of detection, 8 femtomolars. And to put things in kind of terms of PCR assays, basically PCR has a resolution of about 1,000 copies per mil. This particular lower limit of detection of 8 femtomolars corresponds to about 10x that PCR limit of detection, so about 10,000 copies per mil. But it takes us only about 5 minutes to do the detection, whereas with PCR, the thermal cycles to be able to replicate the DNA or the RNA can take about 2 to 4 hours. And especially when we want to look at environmental samples, for example, ocean water or wastewater, inhibition that comes from all of the salts could be a pretty substantial challenge. So beyond just looking at nucleic acids, we wanted to extend this to metabolites. And here we use the same format of basically looking at shifts of our high quality factor resonances when we had a toxin. So in this case, instead of looking at a 20 base pair long nucleic acid fragment, we were looking at a small molecule, microcystin, that is known to be produced by phytoplankton when it's about to enter a harmful algae bloom state. And by directly flowing in the ocean water, we could detect the microcystin toxin. And here we have, with the small molecule, a shift of about, again, 1 nanometer between just adding the linker, the glutaraldehyde, and the microcystin. And then to be sure that we have specific binding, what we can do is flow in an antibody that's known to be specific to this toxin. So the antibody is quite a bit larger. And as we flow in that antibody, now we get about a 2 nanometer shift. So that antibody basically confirms that it was microcystin on the surface that we were detecting. For a lot of these toxins, though, as well as for kind of proteins and metabolites, there are no antibodies that have been developed that can tell you if you're going to be specific to a particular molecule. So what we wanted to do is basically engineer each of these resonators in such a way where we could enable vibrational spectroscopy. So that way we could use the vibrational scattering signature as a fingerprint for what has landed onto the surface. So what we did is re-engineer basically each of the cavity regions in such a way where we have this high quality factor resonance, basically right where the incident laser light is coming in. And then what we do is basically rely on the vibrational scattering fingerprint, or in this case, the Raman scattering signature. So we take the cavity region and engineer into it kind of a broad, in this case, a magnetic dipolar resonance that basically enhances the Raman scattering signature. And we know that Raman basically goes as e to the 4th, where we have to think about both the incident electric field, basically at the pump wavelength, and we square that. And we also have to think about the scattered Raman wavelength, and that basically is that second factor of e squared. And then by kind of revamping the design of each sensor, what we could do is basically put down molecular monolayers, in this case of certain proteins, like an albumin protein or BSA or a streptavidin protein. And now what we can do is get Raman scattering signatures from just a molecular monolayer of these proteins. So now we don't need to have a specific antibody that's developed to be able to tell us if a molecule has landed on the surface. We can use the vibrational scattering signature to tell us what's landed. And here you can see in this black dotted line, the resonator response. So here we have a pretty sharp resonance. I think the quality factor was about 500, so room to improve it here. But you can see off the resonance, there's essentially no Raman scattering signature. But once you tune the laser frequency onto the resonance, now you can see pretty clear Raman scattering. And we can use this vibrational scattering technique basically to determine which molecules have landed, as well as whether those molecules have different functional groups attached to it. So here, for example, is a mucin protein or a mucin peptide. And if you look at the Raman scattering signature of the wild type mucin, you can see it has pretty strong peaks right around 1300 inverse centimeters, as well as about 1450 inverse centimeters and about 1620. And then when you add in a sugar group, basically to 2 different amino acid residues, basically that becomes a glycosylated mucin peptide. And you can see that the glycosylated peptide has that same peak, say right around 1620, but also has a very sharp peak right around 1600 nanometers. Similarly, we have a sharp peak here right around 1100 nanometers. And both of those peaks correspond to the sugar groups that are being added to the chain. Similarly, what you can do is look at different post-translational modifications on a different peptide sequence, in this case on an ovalbumin epitope called synfecal, where here each of the letters corresponds to a different amino acid. And basically, if you acetylate a particular amino acid, like the S residue or the K residue, or if you phosphorylate the S residue, or if you glycosylate the N amino acid residue, you can see the vibrational scattering signature all change in subtle ways. It's actually pretty hard by eye to see exactly how the spectra is changing. But what we can do is, across our many resonators, collect dozens to hundreds of spectra, and then essentially compress those spectra using something like a principal component analysis, or in this case, it's a t-SNE analysis, kind of a nonlinear PCA or UMAT. And basically here, each dot corresponds to a spectra. So we've kind of featurized each spectra, and you can see that the glycosylated version of the peptide all kind of groups together. And that grouping is quite separated, for example, from the acetylated version of the peptide that has just one residue, this K amino acid acetylated. But as you might expect for a principal component analysis, those molecules that are most similar to each other are spaced a little more closely. So basically all of the acetylated versions of the peptide or the molecule are more close together than the versions that are not acetylated. Okay, so that was kind of a nice fundamental study into how we could use vibrational scattering signatures to detect various molecules in a label-free fashion. What I want to share with you is basically how we can use now this label-free format to detect pathogens in wastewater. And probably many of you have experienced during the pandemic how wastewater levels can be used to track infection. So this is an emerging area of epidemiology known as wastewater-based epidemiology. And it's actually quite easy to use PCR to monitor specific viruses in wastewater. But it's really hard to use PCR to be able to apply it to the broad array of bacterial pathogens that could be present. And that's because there are of order a few dozens of bacteria that might be present. And if you were to try to detect all of them by using different PCR primers and thermal amplification, usually inhibition would either reduce your sensitivity or your specificity. So we wanted to use, again, this Raman scattering technique, in this case, though, not on individual biomolecules, but instead on cells. So if we take a cell and we illuminate it with a laser, we know that Raman scattering gives us basically red-shifted fingerprints of the cell or the molecule that we're going to look at. So what we did is collect single-cell bacterial Raman spectra from 22 species and 9 strains of bacteria, and these represent 95% of all patient cultures at Stanford Hospital. And by collecting the Raman scattering signatures from individual cells, you can see that the Raman signatures are quite different between certain strains. So for example, Pseudomonas pneumonia or S. pneumonia has a very different scattering signature from E. coli, whereas other strains have more similar Raman scattering spectra. So you can see, for example, that MRSA and MSSA, the methicillin resistant versus susceptible strains of Staph aureus, look more similar. And what we did is basically train a neural network that allowed us to feed in this case, over 60,000 single cell spectra for these different bacterial species and strains. And then what we did is test it on over 12,400 spectra, basically using the Raman as a proxy for essentially mass spectrometry. And we know the ground truth for mass spec, the advantage of Raman being that it's much higher throughput and also much faster. Usually we just need about 15-second acquisitions to be able to determine what type of cell it is. So here, what comes out of our neural network is essentially a confusion matrix where we know the ground truth for mass spec, and then we can say what the neural net predicts based on our Raman spectra. And for the most part, you can see that we're getting appropriate classification. So we have most of our predictions agreeing with what the ground truth is. These are reported in percentiles. If there are misclassifications, those are reported as percentiles that are off the diagonal. So you can see sometimes it misclassifies this particular strain of Staph aureus as a methicillin susceptible version of Staph aureus. And if we zoom out to the full 31 species and strains that we looked at, you can see overall we do quite well. The average strain level accuracy is about 82.5%, which is not too bad given that we were training it on a relatively limited data set, like I said, about 60,000 single cell spectra. I think one of the cool things is that beyond just strain level accuracy, what we can do is kind of group each of these pathogens by first choice antibiotic. So basically, if a physician were to see a particular bacteria, they would want to treat the bacteria with an antibiotic based on, for example, like the geographic region or maybe the age of the patient. And basically, all of these bacteria are color-coded by the same first choice antibiotic. And you can see that a lot of the misclassifications occur within the antibiotic grouping or the antibiotic treatment group. And the average first choice antibiotic treatment accuracy is 97%, which is actually higher than the accuracy that you would get with mass spec. So then what we did is move from some of the cell line and kind of pure or like clean samples to clinical samples. And these are patients that were known to have tested positive for E. coli or MRSA, MSSA. And what we did is take our cell line spectra and then feed in some of the clinical data. And you can see that initially for these first 30 patients, there were some misclassifications of the treatment. But then once we had fed in this clinical data and we took a second set of 30 patients, once the neural net had already seen some of the clinical data, it actually performed much better. And we were able to get over 99% treatment accuracies by looking at these 60 patients from the Stanford Hospital and the VA. So what we wanted to know is not just could we use the neural net to predict the species and the strain and the first choice antibiotic, but could we actually use it to predict kind of drug resistance? And this is especially important in tuberculosis, where it can take about 40 days to culture the bacteria. So usually it's very easy in the global south to identify the presence of tuberculosis in a sputum sample or potentially in a throat swab. And that's because TB has such a distinctive elongated shape, and it usually is pretty easy to do PCR on the sample and identify which or whether TB is present. But then it takes about 40 days to be able to do the antibiotic susceptibility testing to know which drug to prescribe for the patient. And the reason why that's a problem is because many of you have probably heard that antimicrobial resistance is becoming a global epidemic and is predicted to kill more people than most cancers and AIDS combined by the year 2050. And usually if you test positive for a bacterial infection, in particular TB, you're given broad spectrum antibiotics, and the CDC estimates that at least half of the time, those antibiotics are the wrong type or the wrong dose. So what we wanted to do is basically look at different drug-resistant strains of tuberculosis, where the ground truth had already been confirmed by using traditional antibiotic susceptibility tests, and see to what extent Raman could predict the antibiotic susceptibility. So we have TB, and what we did is grow the TB in a liquid culture with different antibiotics. So amikacin, moxiflacin, isoniazid, rifampicin, and then we also have the wild type TB. And by looking at just the SEMs of each of these cultures, one of which is the wild type on the right, and one of which was resistant to amikacin, you can see that there's no difference basically in the morphology of the cells. So what we did is we picked the cells that we knew were resistant to these different antibiotics, as well as the wild type, and then we collected Raman spectra of individual cells. And we did this over anywhere from 3 to 4 different replicates. So here, for example, we have 4 different types of amikacin, 4 different amikacin resistant pathogens, 4 different types of isoniazid resistant pathogens. And if we just zoom in to the rifampicin resistant pathogens and the wild type, In the Raman spectra, you can see there's not too much of a difference, but there is this pretty substantial peak right around 1050 or so inverse centimeters that actually corresponds to mycolic acid. And one route for TB to acquire resistance is through development of a thicker cell wall or thicker mycolic acid cell walls. So what we did then is collect about 1,000 spectra of individual cells of pathogens that we knew were resistant to moxiflacin, rifampicin, isoniazid, and amikacin. And basically now what we're doing again is that machine learning process where we take each spectra and basically project that spectra onto an individual point. So you can see there's kind of a grouping of these different antibiotic resistant strains based upon which antibiotic it's resistant to. And if we look at the features that are giving rise to classification, what we did is basically remove wave numbers from each of our spectra to be able to figure out which features were most important for classification. And you can see the accuracy of the algorithm dropped down quite a bit when we remove that peak that's right around 1100 inverse centimeters. And that's that CC stretching bond, which corresponds to mycolic acid. We also have important features in the Raman spectra that correspond to a CH2 twist and to a C double bond O stretch. And then if we look at the confusion matrix, you can see with above 87% accuracy, we're able to predict which antibiotic the TB would be resistant to. So I think this is a pretty exciting result that we're getting ready to publish, but it shows that in principle, without culturing and with just a short 15-second acquisition on a microscope slide, we can identify which antibiotics a tuberculosis would be susceptible to within a few minutes of receiving the sample rather than having to wait 40 days. So where we're headed with this work now is taking these chips that have multiple sensors on it and a small footprint, and then enabling multi-target detection. And especially for environmental health, we're excited to be able to look at cyanobacteria, for example, the phytoplankton that are responsible for microcystin-based harmful algae blooms, and be able to quantify not only how many cells are present, but how those cells shed their metabolites into the environment, like microcystin, and then also what DNA is present. So basically, can we look at DNA that's present not only from the cyanobacteria, but also from other organisms in the environment to see how harmful algae blooms are changing ecosystem health? And what we're striving to do in our lab is basically to address the question, who does what under which physical conditions? And then use that information to extend and enhance health spans of life on the planet. And en route to that goal, what we've been doing is working closely with Monterey Bay Aquarium to be able to integrate some of these multiplex sensors onto their autonomous underwater vehicles. And the cool thing is they were already excited to be able to use sensing technologies. They were kind of relying on surface plasmon-based sensors. So they already had a lot of the existing infrastructure where they have this really cool autonomous underwater vehicle that has a lot of the environmental sample processing capabilities already on board. So it has about 100 of these cartridges that basically take the ocean water and filter it down through a series of microfluidics and millifluidics to where the sample is located. And here's now a zoom into our particular sample where we've basically integrated kind of a CCD detector along with our silicon chip on it, and then we can fit it straight into the fluidic processing area, so that way as the ocean water kind of flows over our chip, we can detect cells, genes, and metabolites. And with that, I think we're at the top of the hour, so I'll just summarize by saying that I think nanophotonics has a place in the microbiology lab, and our lab has really tried to combine nanophotonic hardware, in particular silicon photonics, for kind of very rapid reads of biological systems and all the data that's contained with them, with modern software. So because we're getting so much data, we need to find a way to provide the interpretability of that. And what we've done is utilize or capitalize on state-of-the-art machine learning and AI to be able to provide the interpretability to the data we're receiving. And I think the combination of both of these are helping to enable next generation optimization, or at least understanding of personal and planetary health. So I started my talk by sharing the design of these high quality factor metasurfaces, which both confine light into very small cellular to molecular volumes, and they also can direct that light to particular camera positions. And by using bioprinting, we were able to detect over different sensors both nucleic acids and metabolites. And then what we could also do is rely on Raman scattering or vibrational scattering to be able to identify peptides, proteins, as well as bacteria and their drug susceptibility. And I guess in this talk, I mentioned mostly what we were doing with Monterey Bay Aquarium, but we have some really exciting work showing that we can detect bacteria in wastewater samples as well. And in the future, what we're excited to do is basically use these techniques to look at dynamic interactions between, say, pathogens and antibiotics, or between various drugs and proteins. And then we're also excited to enable what we would call CubeSats of the sea. So basically having kind of smaller autonomous sensors that allow us to understand ecosystem health. And with that, I'd like to make sure I thank our funders, especially the NSF, the NIH, the Moore Foundation, and the Gates Foundation, and all of my lab members, both past, present, and future. You got to see many of their pictures throughout my presentation, but I certainly couldn't have done this work without an incredible team of really talented graduate students and postdocs. And thanks to all of you for your attention. Happy to take any questions.
Thank you very much, Jennifer. So yeah, the talk is open for questions. Yeah, yes.
Yeah, I wanted to know for the tapering of the, what was it? For the tapering, why were you using polynomials? I guess I was just curious about that.
Did you hear the
Yeah,
question?
that's a really great question. So yeah, as you saw, we wanted to go from kind of these extended waveguide structures to more confined waveguide structures that would allow for dense multiplexing on the chip. And to do that, we essentially put mirrors at the end of each cavity or at the end of each waveguide. And you can think about kind of different ways of constructing those mirrors. It doesn't necessarily need to be a polynomial. It could be, you know, some other more complicated function. The key is that you want to make sure that light kind of is adiabatically entering the photonic crystal mirror and then being reflected from it. So if you have kind of a potential well or a barrier that's too abrupt, or if you try to confine light too strongly, you'll basically wind up reflecting a lot of light, not so much back into the cavity, but more kind of to other orders. And maybe I'll try to bring up a slide that might better show what happens if you don't appropriately tailor that photonic crystal region. Here, this one might help. Let me see if I can share my screen. So here, the photonic crystal mirrors basically enable the reduced device lengths, and you can think about basically the strength of the mirror as being related to kind of the frequency of the mirror bands. And there is this really cool paper that I'd refer you to. It was published in Nature in 1997. I think one of the main takeaways from this paper is like confine light gently to confine light strongly. And here you can kind of see they have this slab structure that's almost the equivalent of our cavity. And here they have mirrors on each end. And if you put a perfect mirror on there, basically without any tapering or any adiabatic confinement, and then you look at the Fourier transformed electric field, you can see there's actually quite a bit of the field in the leaky region. So you wind up getting quite a bit of radiation that's scattered out of the sides by having too good a mirror. But if you actually adiabatically change how light goes from the slab to the mirror, or basically in our case from the cavity to the photonic crystal, you can design the structure in such a case where you have no light leaking out. So by confining light more gently and having this kind of tapered electric field profile, you're able to get better confinement of the light in the cavity. So hopefully that helps a bit, but if not, I definitely refer you to this kind of oldie but goodie Ferresi Nature paper in 1997.
Thank you.
So I have a question here. Shall I use the mic? So Jennifer, if I understand that you have a sort of array of waveguides, the light is coming from the bottom or top of the of your substrate, and then there's a grading on the waveguide that couples light into the waveguide, and
That's
then
right.
your photonic crystal, the mirrors are basically confining light to increase the Q factor. But then any scattering, you are guiding it towards your sensor because of the spacing between the waveguide is non-uniform or it's controlled somewhere, right? There are two things here. There's grading and there's a spacing between the waveguides that controls it. Am I right with that?
I think that's a good way of thinking about it. I missed a little bit your part about the spacing, but this might explain it a little bit well. So here's the cavity region. And like you said, we basically are varying the width of each neighboring block in such a way so light coming from the top gets coupled into this waveguide region. And then what we do is basically design the mirror region in such a way where we can get kind of maximum reflectivity while not disturbing the dipole-like scattering of the central region.
Okay, thank you. Thank you very much.
Yeah.
Yeah, in line with the question of Govin, Jennifer, I wonder if I understood well, your devices work not only in the plane direction, but also in the perpendicular, in the vertical direction. Is that correct? Do they work in both directions, right? So you can play with both directions in the space to detect things. Okay, that's really very interesting, and I'm also really amazed of the great number of applications you found with you use for this sensor. So it's really amazing what you can do with them and to detect so many things, so very interesting things, not only COVID, but also very much interesting things on other ways. I don't know if there's more. Yeah, yes.
Well, we should take questions in the chat. Are there questions online? Yeah, on chat. There seems to be something.
Chat.
Jennifer, are there questions online on the chat?
Doesn't look like there are any just yet.
Okay.
I like the person who answered the data question though with a zillion gigabytes. I guess you put a good bound. Wait.
Yes, and there's another question here.
Yeah, I have a question about the paper as well. And so it seems that a choice of having a polynomial structuring of the mirrors confine the light gently. So what I'm thinking is that, have you thought about using machine learning to optimize the design of the mirrors?
Yeah, really awesome question. I don't know if you're a student or a postdoc, but feel free to come on over to Stanford if you want to do that. We've just started thinking about how to use kind of ML and evolutionary design to be able to further optimize these structures. Because kind of to first order, we've just used our own electromagnetic insight to be able to make structures that are pretty good. But I think folks who are in this field and have really thought about how to use AI guided photonic design have shown that sometimes it's the most non-intuitive structures that give rise to the best performance. So yeah, we've only just started thinking about that and kind of thinking about which functions we want to optimize for. On the one hand, there's the quality factors, there's also the field distribution, there's also kind of the dipole scattering. But yeah, I'd be really excited to see how we could use AI to guide the design.
Thank you.
Thank you very much. I think that's it. Question? That's it. Well, yeah. So thank you very much, Jennifer, for this great talk today. And thank you very much to all of you for being here today. Yeah, with this, we can say bye to Jennifer. Thank you very much.
Well, thanks to all of you. I see the coffee up there. Make sure you put it to good use. I would definitely be grabbing some if I were in person. I hope you all have a great week.
All right. Thank you very much.
Thanks. Bye.
Thank you.